So switching now to TES, transcranial electrical stimulation. Again, brain stimulation generates some fun historical figures. So that's an early attempt at researcher stimulating his research assistant's brain with very little success, as it turns out. So TES was really first published in the late 1990s, although there were a couple of earlier attempts that didn't really get anywhere. That sort of showed that you could pass a weak electrical current through the brain and get some effects. But it was really a couple of studies on the motor system in like 1998 that really defined TES as we now know it. And so, as I mentioned before, TES involves stimulating the brain by putting electrodes on the head, minimum of 2, because you're passing a current from one to the other, and then passing a very weak current. And so where TMS is tens of amperes, where amperes is a measure of electrical current, TES is 1 to 2 milliamperes. And typical power source for a TES system is a 9-volt battery. So it's a very low level of current, it's very safe, there's no real risk of like burns or anything like that. We'll talk about risks a little later. And you can see that the common system, this is changing a bit, but historically the system that's been used involves fairly large sponge electrodes. And the reason for that is that although this is relatively safe, the current that you pass through does kind of tingle and can feel kind of itchy or uncomfortable to people and actually cause some burning just because of impedance. So the more impedance you have between the electrode and the scalp, that's resistance. Resistance can cause heating, can cause burning. And so by having a larger surface area, you can actually, you're sort of sending the same amount of current through a wider patch of skin, and you get less of these side effects. So that's kind of what the system looks like. And one of those electrodes is defined as an anode, and one is the cathode. And the current is basically flowing in through the anode and out through the cathode. So that's the sort of basic effect. Now, as I mentioned, there have been some changes. So the newer mode is what's called HD, or high definition TES, which involves smaller electrodes and more of them. And in this case, you're getting around that issue of the needing the bigger surface area to reduce the discomfort by actually using, instead of one anode, you can have multiple anodes or multiple cathodes. And so you're basically distributing the current flow over a larger area. So through multiple electrodes rather than through one big electrode. And in these cases, often you'll have software that allows you to actually target brain areas, so kind of a neuronavigation type system. And so you can design the positions of the electrodes and the strength of current flowing through each individual electrode to more precisely target a particular brain area. And I'll come back to why that's important. I mean, it's sort of intuitively important, but it's actually very important, it turns out. So I'll come back to that a little later. So the basic effect where you have one anode and one cathode, which is kind of how we'll talk about things for most of the rest of the lecture, is that you get increased neuronal excitability or sort of stimulation effects at the anode and decreased neuronal excitability or inhibition type effects at the cathode. And these seem to be relying on modulating the membrane potentials of neurons and basically sort of biasing them in one direction or the other. And so one session and a session will typically last like 5 to 20 minutes of stimulation itself. And the effect of that can last for as little as 5 minutes and up to an hour and a half after the stimulation. And like TMS, there are approaches that have been tried for clinical treatment type purposes where people have many sessions, like a session every day for 10 days. And those seem to have effects that last more than a month. So they can have, again, ongoing sort of, seem to induce neuroplastic changes that are lasting well beyond the period of stimulation itself. And broadly speaking, we can talk about online TMS studies where you're looking at what's going on, measuring behavior as people are being stimulated, or you can look at that period afterwards, however many minutes, 5 to 90 minute period afterwards, and look at the lasting effects of that, and also use that to sort of assess how long the effects last for. As I said, multiple sessions over multiple days. And just like TMS, there are different flavors or protocols for TES. They're totally different from the TMS ones, however. The simplest one and the most widely used one is tDCS, or transcranial direct current stimulation. In this mode, you just turn the current on and you leave it on at the same level for the entire period, and then you turn it off. And so what you're seeing here is the anode, the current is positive, so in this case 2 milliamps, and at the cathode it's the opposite. It's negative 2 milliamps. tACS, or alternating current stimulation, involves an oscillating sort of sine wave type stimulation with some frequency. Usually the frequencies are comparable to human EEG frequency bands, so like alpha is 8 to 12 or 8 to 14 hertz. So you could stimulate in the alpha range or the gamma range or something like that. Critically, it oscillates at both the anode and the cathode, and because it's oscillating positive to negative at both, there's no real functional distinction. So you get similar effects of tACS at both the anode and the cathode. A variant of that is oscillatory tDCS, where it's still oscillating, but you adjust things such that rather than oscillating above and below zero, as you see in tACS, it's all above zero at the anode and all below zero at the cathode. So you do get different effects at the anode and the cathode. This one is relatively rare. I sort of struggled to actually find papers that were using this, so it's not very well understood. tACS is much more widely used, and I'll go into a bit more detail on each of these. And then TRNS is random noise stimulation, and with this you're basically stimulating at a random strength on a sort of sample by sample, where your sampling rate is maybe 1,000 hertz. So 1,000 times a second you're sampling, or you're stimulating at different current strengths just randomly. And the effect of that is, if you look at that over time, it's kind of like almost like white noise. So you've got multiple different frequencies that are occurring. So you've got some level of stimulation at like 1 hertz, 2 hertz, 10 hertz, 20 hertz, typically up to like 100 or more hertz. So it's doing a lot of different stuff. So tDCS, as I mentioned, is the oldest and most widely used effect or protocol for stimulation, and it tends to have the excitatory effects at the anode, inhibitory effects at the cathode. And one of the interesting things is that while you can sort of get increasing effects over a 5 to 13 minute period, after 20 minutes it seems like the effects actually reverse, and so the anodal stimulation causes decreased activation in that area. So as an example of where tDCS is used, it's often used in working memory studies, also in the motor system, but in working memory, you stimulate over prefrontal, dorsolateral prefrontal cortex areas that are known to be involved in working memory, and anodal stimulation over that left dorsolateral prefrontal cortex has been shown in a number of studies to actually improve people's working memory. And so it's like you're biasing the effects or the currents, as neurons are a little more likely to fire, and people are better at working memory. But if you stimulate for more than 20 minutes, their working memory performance will actually go down. And it seems like that's maybe just you're sort of overstimulating the neurons, and they kind of colloquially get exhausted, and so the effects are different. So there's limits on how much you can do. This is the original study by Nishi and Palos. Sorry, it's 2000, so not quite the original. I think they had a 98 or 99. But very similar to what I was showing you with the TMS motor mapping, they were doing anodal stimulation over the motor cortex, and the cathode would then be over the forehead. So the idea was that sort of a neutral part of the brain not involved in motor function, and looking at motor evoked potentials as a function of the stimulation. And they showed that the MEP amplitude went up with anodal stimulation over the motor cortex. When you reverse that and the cathode is over the motor cortex, you actually get a suppression of the motor evoked potential relative to here. The dashed line is the baseline, so that's with no tDCS. That's kind of the level of your motor evoked potential. So again, you get anodal stimulation, cathodal inhibition. The other thing which I sort of extracted and highlighted in the paper there was that even at a relatively low level of stimulation, it's 0.4 milliamperes. So remember I said that the normal level is like 1 to 2 milliamps. So at 0.4, people experienced itching stimulation at both electrodes and possibly flashes of light. And this is important because, again, we have this issue of blinding, and does the subject know that they're being stimulated? Yes. This is a very detectable kind of thing. especially with the direct current stimulation. As you're ramping that up, they start to feel this weird sensation. It's not necessarily present during that steady state, during most of the stimulation period, but the ramp up and the ramp down, people do seem to be able to feel that. So people have done things as a control, like ramp it up and then ramp it back down right away, so that you feel that tickling sensation. You think you're being stimulated, but during most of the period, you're actually not being stimulated. I'll come back to controls later. So tDCS has effects like enhanced motor evoked potentials, as I showed you. Again, over motor cortex, you can get faster reaction times on tasks requiring motor responses. Cognitive enhancement, including again, faster reaction times in memory tasks, improved accuracy on sort of decision tasks, improved memory accuracy. And there's actually these sort of combined effects that if you give people tDCS over working memory areas while also training their working memory using like cognitive training protocols, you can get amplified effects that are greater than either tDCS or the behavioral cognitive training on its own. So there seems to be some super additivity there, which is very interesting from like a cognitive enhancement and a therapeutic kind of perspective. With tACS, typically what people are doing is actually trying to what we call entrain innate EEG rhythms. And so again, you've got things like your alpha rhythm, and so rather than just sort of randomly coming up with some frequency of oscillation, you say we want to look at the effects of amplifying the alpha rhythm in this particular brain area. And so you'll send in your oscillating current at something in the alpha frequency. And what works best is you first put EEG on the person's head, measure what their alpha frequency rate is, because different people, it might be 10 hertz, it might be 11 hertz, whatever. So you measure what their actual alpha frequency is, then you deliver tACS at that frequency, and you can really amplify the alpha that they get. And if that particular frequency, and I'll show you an example of this in a minute, but if that frequency is associated with like some sort of cognitive task performance, you can again enhance that cognitive task performance by basically sort of entraining and boosting the oscillations at that target frequency. And the interesting thing is that's not just during the stimulation period, but actually after the stimulation period as well. And as I mentioned, this is occurring at both the anode and the cathode. So using this approach, you can actually induce oscillations in two areas at once. You can sort of drive two areas to oscillate at the same frequency, and then explore if that improves task performance, which suggests that synchronization in a particular frequency band actually is related to cognitive performance of whatever task you're looking at. One thing that people do actually, even with the tDCS, also with this, is if you really only wanted to target one brain area, the other electrode doesn't have to be on the head. It could actually be like on the shoulder or something like that. And so with that, especially in some cases, you want to stimulate one area with the anode, but you don't, because the cathode causes some sort of suppression, you don't necessarily want to randomly suppress some other part of the brain, even if it's like the frontal lobe or something you think is not involved, because then it's really not a well-controlled experiment. You don't really know if the effects are purely due to the anodal stimulation or the cathodal inhibition. And so if you put the cathode somewhere else on the body that's not on the brain, you sort of get rid of that, and you're isolating that there's only one part of the brain that's being stimulated. So similarly here, if you really only wanted to do tACS at one part of the brain, you just put the cathode somewhere else on the body, and then you can be more specific that way. Another thing you can do is actually, so going back, one thing that's not super obvious unless you look really closely is that when it's, because the current's flowing from one to the other, when the current is maximal positive at the anode, it's actually maximally negative at the cathode. So they're oscillating at the same frequency, but opposite polarity, meaning the timing of the peaks. If you want two areas to be oscillating at the same frequency and with their peaks aligned, you can actually have two anodes and one cathode and do it that way, and that way you can synchronize two different brain areas more directly. So effects of tACS are similar to tDCS in terms of you can do things like improve memory, improve motor learning. You can actually induce or alter sensory perceptions. to the extent that those are associated with these sort of oscillations. And critically, they've been used to demonstrate the causal role of oscillations in cognition. And so for a long time, people have measured oscillatory activity using EEG and made the inference that those oscillations are directly sort of representative of the cognitive activity and sort of necessary for it. But there's no guarantee when you just sort of have people do something and measure their brain activity that there is a direct causal link. And so with tACS, you can actually boost particular oscillations, and if that actually boosts people's performance or perception or whatever, that gives you more of a causal link. And similarly, if you can actually sort of suppress that, performance goes down, that also suggests a causal link. I thought this was a very cool study, and also this is actually a figure from their paper, which is like way nicer than most of the figures you find in science papers, and quite effective, if a little gratuitous at times, but that's okay. So what they were looking at here was based on animal studies showing that in working memory, there's actually two frequency bands that are important. One is theta, which is a slow, low frequency, so like a 2 to 4, 4 to 7 hertz, 4 to 7, I think. So a relatively slow oscillation. And then gamma bursts that typically occur at the positive peaks of the theta waves. So you can see the positive there, and then the trough. So the gamma burst, which is a higher frequency, like 40 hertz or above, occurs specifically on the peaks of the theta and not at other times. And the gamma bursts seem to correlate with the number of items that are being held in working memory. And so again, the researchers were interested in whether there was a causal link between the timing of the gamma bursts and working memory performance. So they applied tACS to left dorsal prefrontal cortex, so working memory area, and tested people's working memory performance and showed that when they simply just did theta entrainment and boosted the theta, people did show an improvement on working memory. They were also looking at this EEG connectivity index, which we can worry less about. When they just did the gamma bursts, they didn't actually affect working memory performance. But when they did the gamma bursts on the theta peak, which is the effect that was being observed in animals, they actually boosted memory performance even above, so like maybe not double, but significantly more than when they just did the theta. So there's some involvement of both the theta and the gamma in there, and specifically timing the gamma with the peaks of the theta, because they were also able to time the gamma with the trough of the theta, because they're controlling everything, and there they found no effects on working memory. So again, it shows a nice causal link that, okay, we sort of see this occurring in the wild, but does it really make a difference? So we can actually sort of turn it on and turn it off or amplify it and show that that has a direct causal link to people's working memory performance. So kind of a nice example of how you can use that. And that's more and more the way people are thinking of tACS, is tying it to oscillatory activity with EEG and looking at those two together. The random noise stimulation, I already sort of described how it works. Typically contains a range of frequencies from very low to very high, and some studies have compared that and shown that you get stronger effects with if you just do the high relative to just doing the low. People don't really understand why it's working yet. It doesn't seem to be just incidental tACS, meaning that because there's all these different oscillations in there, if a task relies on, say, alpha or theta or whatever, there's some amount of alpha and theta entrainment going on with this. And so initially people thought, well, it's just you're stimulating at all frequencies, so any frequency that's involved in brain activity is going to get a boost. But it doesn't seem to be that. It seems to be a fundamentally different mechanism. And in fact, again, relative to tDCS is by far the most widely used, tACS the next most widely used. tRNS, there's far fewer studies, but the ones that have been done, especially comparing to tDCS, seem to show that tRNS has similar effects, but stronger. So this is an example from, this was a line orientation judgment task. So you can see, you saw one line and then a second line, and you just had to say if they were parallel or not. And people's performance, this is over blocks, so basically everybody got better with practice. But the top line here, so this is performance. The best performance was with the high frequency tRNS. Next was with the low frequency tRNS. And then ANDL tDCS is the dotted line here. and you can kind of see the confidence intervals. And so basically the tRNS had dramatically stronger effects on performance than the tDCS. So it seems like it might have similar but stronger effects. It's been found in memory tasks as well. So it seems quite promising, but people haven't done it as much, so it's not well understood, either what it's doing or how well it works, or if it's as consistently better than tDCS, as is suggested by the few studies that have been done. Broadly speaking, these are modulated by the kinds of neurotransmitter systems and ions that you would expect. So calcium and sodium are critical for controlling the balance of electrical potential inside and outside the cell. And if you block or interfere with sodium or calcium, then you get reduced impact of tDCS or TES, broadly speaking. There's also effect of glutamate and GABA, so excitatory and inhibitory neurotransmitters, and kind of in the direction you would expect, although weaker effects at the cathode than at the anode. And so broadly speaking, the models of how this works, the initial models were very descriptive. So this AECI, or anodal excitation cathodal inhibition, suggests that basically you're just sort of, by stimulating at the anode, you're causing the electrical potentials of the cell membranes to be a bit more likely to fire. So you're sort of biasing them a bit in the direction of depolarizing, so action potentials are more likely to happen. And if action potentials happen, that's more brain activity in that area. And so you're sort of just increasing, without driving the brain activity directly, you're making it more likely that those cells will respond to input. And so you're more likely to fire, which could give you like a bigger motor evoked potential. In terms of how that explains better learning, like better working memory performance or whatever, if the cells are more likely to fire in response to input, that's your basic Hebbian LTP mechanism, right? As one cell fires, and then the receiving cell fires in response, and that strengthens the connection between them. So if you bias those receiving cells to be more likely to fire, then you're going to increase the likelihood of LTP, so things like memory performance and so on should improve. So that's the sort of simple model. The activity-dependent model just builds on that and says that if the neurons are already depolarized, they're already more likely to fire for any sort of reason, like they're actively engaged in a task, or you've recently practiced a task. The stimulation can also sort of drive that. There are some interesting sort of what we might call paradoxical effects, though, which is that on hard tasks, tDCS seems to improve learning, but on tasks where people are already good, it doesn't actually seem to improve, and in some cases can actually hurt with learning. And similarly, in working memory, tDCS had a much bigger benefit for people who had a low baseline of working memory performance relative to those who had a high baseline. And so it suggests that it can provide some boost, but if the brain area is already functioning at sort of an optimal or high level, the effects of the stimulation might not be beneficial, and they might actually be disruptive, because if you're sort of already at the sweet spot of brain activity, if you drive that brain activity to be higher, you might just be creating more noise in the system rather than positive change, right? So if the neurons have to sort of be stimulated to a certain level to fire, and they're already there, then the extra input is just going to cause them to spontaneously fire more, and maybe sort of fire in response to inputs that they shouldn't be responding to. And so you're going to impair learning or performance just because of that. And so that's what's captured in this diagram here. So this was a study where the researchers were looking at people's sensitivity to moving dots. And you can do this in a paradigm where you've got lots of dots and they're either all moving in random directions, or there's some level of coherence, meaning some proportion of the dots are all moving in the same direction and the rest are moving in random directions. So it could be like 40% of the dots are all moving to the left, and the other 60% are all moving in different directions. And so you can use this in a very simple standard kind of psychophysics paradigm where you manipulate the proportion of dots moving in a coherent direction, and as a baseline just determine what proportion of dots has to be moving in one direction for people to be able to consciously detect it and press a button, right? And then you can apply TES to, say, V5, our motion-sensitive cortex, and see if that improves or impairs their ability to detect those dots. And so this is showing here random directions of motion. And this is basically the firing response of neurons that are tuned to different directions of motion, because different neurons are tuned to different directions of motion, right? So with random directions of motion, these things are all getting stimulated to some way. And after TES, you basically just sort of amp up everything, right? So, that's kind of a sort of baseline state to look at, because they're all moving in random directions, so you can't make a judgment of what direction they're moving in. Now, if there's partial coherence but relatively weak, say only 10% of the dots are moving in one direction, that's a very hard task, and most people are not going to be able to tell which the direction of coherent motion is. And so the notion here is that one population of neurons is being stimulated by the dots a bit more than other ones, but it's too weak to hit the sort of conscious detection threshold, which is that dashed line there. And so if you add a certain level of electrical stimulation, make those neurons a bit more likely to fire, then you're going to bump everything up, but there's sort of a sweet spot where you're bumping up the neurons in the coherent direction enough that that becomes conscious, but everything else is still sort of below the threshold. And so you get better performance in that case. But you can get this paradoxical effect where performance goes down if, say in this case, the stimulus is already detectable. You add in the electrical stimulation, you basically increase the noise. And so all those other directions of motion are now causing stronger firing rates in these neurons. And so the task actually becomes harder for the person, because now all those other direction neurons are firing more. They're firing above threshold, and so the person gets confused and they can't detect that as well. So it's not like just that more stimulation is always better. In this model, the idea is that you're sort of boosting the probability that things are firing, and there's kind of an optimal level at which that gives you better performance. But once you exceed that optimal level for whatever reason, because you were already there or because you're increasing the stimulation too much, then performance goes down again. And this is interesting because one of the big controversies in the literature, I'm aware of the time, I'll let you guys go in just a minute. The biggest controversy in the TES literature is that the results haven't been consistent from paper to paper. And so like with the working memory stuff, there was a review paper that basically reviewed all the working memory studies using TES up to that point and showed that on average, people didn't actually show improved working memory with TES. Other papers came along and said, yeah, but you didn't really look into their methods, and some of them had better methods than other ones. But that's one of the factors, right, is are you sort of in this situation where you're taking people and you're sort of boosting them to a level of working memory performance that's a little bit better and they weren't strong already, or are you taking people who already have strong working performance and actually causing worse performance because you're going over, right? So that's one of the considerations. The other is this, which is it turns out when you model how current flows through the brain, when you use these big sponge pads and just apply them to somebody's random brain, the current, first of all, you get huge swaths of the cortex being activated and not consistently directly under those sponges. And even at the cathode, you can get weird sort of not only inhibitory but sometimes excitatory effects. And so that's what's really motivated the HD TES, is that when you have the anodes surrounded by cathodes over a distance of only a couple of centimeters, you get, and you model that, you get very focal stimulation around the cortex. But even then, it's sort of, and this is a zoomed in, you can see a sulcus there and gyri on either side. You can actually get sort of peaks and troughs of stimulation strengths in a very local area. And so basically what this paper argued for is that you can't just blindly stick big electrodes on each individual's head and expect you're going to get consistent results. You need to actually model the current flow and design the array of electrodes and strength of stimulation around that person's neuroanatomy and target it, and then you're going to get more specific effects. Even then, and I think this is the last slide, studies have shown that in some studies at least, only about 50% of people show the expected effects of TES. And the rest are either non-responders or even sort of reverse responders. And this seems to vary on a lot of things like skull thickness, neuroanatomy, how the things conduct, baseline levels of activity, as we've already talked about, any medications they might be taking, or even things like nicotine or caffeine. And in women, the responsivity seems to vary over the menstrual cycle as a function of different hormones. And so there's increased awareness that we're sort of going from a phase where it's just like, let's stick these big electrodes on and everybody shows magical effects. Or in fact, what was happening was the people who got the really cool effects got published, and all the people who weren't getting it were really frustrated, but they just weren't publishing. So there wasn't really a good estimate of the actual effectiveness of tDCS. And so it's studies like this where people are now understanding, it's actually a little more subtle. We really need to understand this better and be more careful and systematic about our methods. So that's kind of the take-home message, I guess, for TS is read the method section and think hard about what's being done there. And that's where I'm going to leave it. Thanks for your attention.